Today's lecture on hybridization technique or procedure. A successful hybridization requires a different procedure. The procedure includes some important steps. These steps are as follows: that is the selection of parents, selfing of parents, hybridization technique, harvesting the hybrid seeds and raising F1 generation. To apply a proper selection method, we will see one by one later on. First of all, we will see the selection of parents. The selection of parent is the most important aspect because the character in the parent will be available in the combined form in hybrid. While selecting the parents, there are some important characters are observed, that is the agronomic base, genetic diversity, complementary nature of male parent, homozygosity in both parents, and combining ability. Then selfing of parents, the pure line procedure produce a better variety due to elimination of undesirable characters, and such a pure line and their hybrid can be maintained easily. For obtaining the pure lines, selfing in both parents is essential. The artificial self pollination or selfing, that is the removal of undesirable characters and retain two breeding characters. By repeating a selfing, homozygous nature and true breeding nature of many required characters in the parent can be achieved. The homozygous parents are then testing for their combining ability. Hybridization technique. This is an important part in the entire process of hybridization. All steps in this technique are to be carried out carefully and in a proper sequence. That is the isolation of selected and self parents, emasculation, bagging, tagging, then artificial pollination, then collection of seeds, selfing in F1, and proper selection method. And isolation of selected and self parents. The selected plants, after testing their combining abilities, they are grown separately on a separate plots to avoid a cross pollination. For completion of remaining steps in the procedure, both parents should be exhibits a simultaneous flowering under the natural conditions. If they are unable to produce flowers at the same time, by adjusting the time of seed sowing or by giving a photoperiodic treatment, the parental lines should be made to flower simultaneously or at the same time. Then we'll see the emasculation, that is the removal of anthers from a flower bud before anthesis, that means the dispersal of pollen. It is called as emasculation. This is essentially in bisexual flowers on a female parent. The emasculation can be carried out by following different methods. The first, that is the physical removal by hand. In a large flowers like cotton, the anthers can be removed physically by hand. Then second one, that is the forceps or scissor method. If the flowers are relatively small and produced in groups, the anthers cannot be removed by hands. In such a plants, use a small instruments like a forceps and a scissor, for example in a maize. Then another method, that is the hot water treatment method. When the flowers are very small and use of instrument like forceps and scissor is not possible, at that time the hot water treatment is followed. The temperature of the water is between 45 degrees to 48 degrees centigrade. It is used for killing the pollens. The time required for the treatment varied from species to species. Generally, it is 1 to 10 minutes. Then another method, that is the cold water or chilling treatment. In some plants, the cold water is used for killing the pollens. For example, in wheat, the temperature is maintained near the freezing point. Then another method is also used, that is alcohol treatment. Instead of water, the organic solvents like alcohol is used in some cases. Generally, 55 to 60% alcohol is used for 1 to 10 minutes for killing the pollens. Then another method, that is the genetical method. The use of female parent, which is genetically male sterile, is the best method and it does not require emasculation because in such flowers there is no development of stamens and pollens. Then after emasculation, the bagging is essential. That means the emasculated flowers used as a female parents are then covered with polythene bag or a paper bags. It is called as bagging. It is essential to prevent the entry of any foreign or unwanted pollen grains on a stigma. The bagging should be carried out before stigma attains maturity and becomes receptive. The male flowers are used as male parents and are also covered with paper bags to avoid any contamination in the form of foreign pollen grains. In such a covered flowers, the ripening of the stigma in the female flower should be a match with the time of anthesis in a male flowers. When both the processes, the synchronized successful pollination can be achieved. Then another step, that is the tagging. After bagging the female and male flowers, to every bag covering with the flower label or tag is to be attached. On this label, the necessary information regarding the date of emasculation, name of variety is to be written. The label should be a brief but complete and should specify the name of parents and numbers referring the entry in the field book. Then next step, that is the pollination or crossing. This process consists of two steps. That is the collection of pollens from the female parental flower and transfer of collected pollens to a receptive surface of stigma in the female flower. The pollens are collected only after the natural dehiscence of anthers in a male flowers. The mass of the pollens collected in a petri dish or a small paper bag or by vacuum suction. During the transfer, the pollens for the pollination, the first bag on the female parent is removed temporarily and the stigmatic surface is dusted with pollen mass collected in the bag or in a petri dish. This process completed as quickly as possible. Immediately, the female flower is covered with a removed polythene bag or a paper bags. This artificial pollination is to be completed within the viability of the pollen grains collected from the male flowers. For dusting the collected pollens, the camel hair brushes are used. Sometimes, instead of collecting the pollen, dehisced anthers is picked up with the forceps and rubbed on the stigmatic surface directly. After covering the female flowers with the pollens on the stigma, the date of crossing is mentioned on the label. The label is to be kept to be till the seed setting is completed. It should be with all information and tied properly to the bag. Then next step, that is the collection of seeds. The seeds collected after they complete the growth and mature. The seeds are properly preserved and used to raise the F1 generation. Then the selfing of F1 plants. The F1 plants raised from the collected seeds are allowed to undergo the self-pollination and the seeds for the F2 generations are collected. Then proper selection method. From F2 generation onwards, by proper selection, these individual seeds giving a high yield and other desirable characters are collected and multiplied. This new variety then certified and under the new name distributed to the farmers to cultivation. Then we'll see the another part, that is the objectives of hybridization. It is the first objective to combine the desirable characters of two varieties or two species or two genres. Then second objective, that is to remove the bottleneck genes, that means to remove an undesirable characters like a disease susceptibility. This can be achieved by selecting the parents with a very little difference in a genotype and its phenotypic characters. Then third objective to develop a variety with many desirable characters and overall improvement in the existing varieties. The fourth is to increase the range of genetic variability by introducing different recombinations of characters. Then another, that is to exploit and utilize the hybrid vigor to increase. Then we will say the merits of hybridization. The first merit, it helps to create the heritable variations which are useful for evolution in a species, or it add a new species or a variety. Then second merit, superior varieties can be developed as the process helps to combine all useful or desirable characters into a single variety, generally shows a hybrid vigor. Then third merit, that the hybrid variety is useful to increase the crop yield as the disease resistance, drought resistance and other such genetical characters combined with a high productivity character. Then fourth object merit, that is the new hybrid variety may show adaptation to various environmental conditions, so it can be cultivated in a different regions having a different atmospheric and soil conditions. Then we'll see the demerits of hybridization. It is a time consuming and expensive method. It is expensive because the technical persons with skilled and unskilled labors are to be employed. The time consuming nature is due to confirmation of variety with desirable characters only after third generation. This program can be carried out only by experienced plant breeders and only at experimental stations. Ordinary farmers without technical knowledge and equipments cannot carry out the experimental part in the hybridization process. Another demerit is the success percentage in the process is very less due to difference in the genetical nature of parents. For this reason, a variety in any crop with all desirable good characters is yet to be developed. Thank you.